We are so much privileged to be here. And in this session, we are going to look at Plasmodium life cycle, or you can call it malaria life cycle. We want to see how the Plasmodium species cause malaria in man and how do they ensure continuity. And if you're new on this channel, please remember to subscribe if it is your first time. Like the video and share to many others. And the life cycle of malaria, it occurs in both man and also in an arthropod known as the female Anopheles mosquito, whereby man is the definitive host. So we are going to see that man acts as an intermediate. Man is an intermediate host of the malaria parasite, whereas the female Anopheles mosquito is the definitive. This is the definitive host. Definitive host means the sexual reproduction takes place in the female Anopheles mosquito, whereas the asexual occurs in man. That's why we call it man an intermediate host of malaria life cycle. And this life cycle occurs in two ways or in two processes. That is asexual in man and then sexual in female Anopheles mosquito, whereby asexual is known as schizogonal stage, which occurs also in two ways. That is pre-erythrocytic, from the word erythrocytes. Pre-erythrocytic means before the red blood cells. And also it occurs as erythrocytic, whereby erythrocytic, this stage occurs within red blood cells. So those are the two ways, or a sexual reproduction, which we call schizogony, is divided into pre-erythrocytic and erythrocytic, whereby pre-erythrocytic occurs majorly in the liver cells, whereas erythrocytic occurs in the RBCs. Then finally, we shall look at the sexual, which we are calling sprogony, which occurs in the definitive host, that is female Anopheles mosquito, whereby we shall see formation of the zygote and so forth. So we are saying the life cycle begins when a female Anopheles mosquito, if this is a female Anopheles mosquito, is biting man, when it is taking a blood meal, the female Anopheles mosquito, and this female Anopheles mosquito should be infective. Being infective, it means it contains what we call sporozoites. These sporozoites are found in the salivary glands of the infective female Anopheles mosquito. So when this mosquito bites man, when it is taking a blood meal, it is going to inject what we call the sporozoites. And these sporozoites injected at the site of bite, they are transported by blood and they go to the liver. And once these sporozoites enter the liver, they enter into the liver cells, which we call the liver hepatocytes, the parenchymal cells of the liver hepatocytes. And when these sporozoites enter, they are going to mature. So these are the sporozoites in the liver, transported by blood. And after reaching in the liver cells, they transform into what we call schizons. And these schizons contains multiple merozoites. So it undergoes division to form multiple merozoites. So this is what we are calling a liver schizont, formed when the sporozoites reach in the liver. And these schizons, we are saying they contain what we call multiple merozoites. So these are the merozoites within. So a one schizont can contain between 8 to 24 merozoites, depending on the species. And we know we have majorly 5 different species of Plasmodium. We are going to see species of Plasmodium such as Plasmodium falciparum, that is PF, and Plasmodium malariae. For them, once they reach in the liver cells, they form schizons, and schizons contain merozoites, and once they mature, it ruptures together with the liver, releasing what we call the merozoites in blood saturation. So once these merozoites mature, we see the liver cell together with the schizons, they rupture, and after rupturing, they release what we call the merozoites. And when these merozoites are released, we said they are multiple, they enter into what we call blood saturation. So these are the merozoites released into, this is the blood vessel, and within this blood vessel, we have blood, and that's where merozoites are released. And this one takes between 1 to 2 weeks for these, depending on the species. Some can take 6 days, others 10 days. So depending on the species, it takes an average of 1 to 2 weeks for these merozoites to be released into blood saturation. But there are other types of species of, so P. falciparum, P. malariae, and sometimes P. norisci. For them, they do not undergo dormant stages. Immediately when the sporozoites reach in the liver, mature into schizons, schizons containing multiple merozoites, whereby when they rupture, they release these merozoites into blood saturation. But there are other 2 species, that is Plasmodium vivax and Plasmodium ovale. So for the sake of Plasmodium vivax and Plasmodium ovale, these 2 types of Plasmodium, for them, they undergo what we call relapse. They undergo what we call relapse, whereby when the sporozoites reach in the liver cell, they undergo into what we call dormant stages. It undergoes into dormant stages, and this dormant stage is known as hypnozoite. So they undergo what we call dormant stage known as hypnozoite. So hypnozoite, these are the dormant stage seen only in Plasmodium vivax and Plasmodium ovale. So these dormant stages known as the hypnozoites, they can take between months to years, and later they can be activated to form schizons, schizons containing merozoites, and when they mature, they rupture, releasing merozoites into blood saturation. That is what we can see. So we are seeing relapse occurring specifically in P. vivax and P. ovale. And this relapse is different from the term known as recrudescence. With recrudescence is due to incomplete treatment by malaria disease. So when someone is suffering from malaria and they are given incomplete treatment, there can be reoccurring of the malaria. And this is what we are calling recrudescence. When someone has not taken the full dose, they can get episodes of attack of malaria, which is the recrudescence. So that is the difference between relapse and recrudescence. In the relapse, we are seeing it is in P. vivax and ovale, whereby the sporozoites in the liver undergo dormant stages known as hypnozoites, and they can be reactivated to become schizons within months to years, hence someone is suffering from malaria. Whereas recrudescence is due to incomplete dosage. When someone is on treatment, does not complete dose, malaria can reoccur, getting episodes of attack by a phenomenon known as recrudescence. So this is what marks the end of pre-erythrocytic schizogony, which occurs in the liver. So once the schizons rupture together with the liver cell to release the merozoites, when they reach in blood saturation, we are now going to start, they are taken and they go into what we call the RBCs. And this one marks the beginning of erythrocytic schizogony, whereby these merozoites attach on the receptors of the RBCs and they transform into what we call ring forms. They transform into what we call the ring forms. The ring forms are known as trophozoites. So they form what we call trophozoites. Those are the ring forms after entering into the RBCs. So we are seeing when the merozoites are released in blood saturation, beginning with erythrocytic schizogony, the merozoites attach on the receptors of RBCs and enter inside the RBC and form what we are calling the ring forms. And ring forms are called the trophozoites, and these trophozoites begin to mature by thickening. They begin thickening. That is the late stage of the trophozoites, which were the ring forms. And as these trophozoites are maturing, they feed on the hemoglobin of the RBC, forming toxic substances known as the hemozoid after feeding on the hemoglobin. So they produce toxic substance known as the hemozoid. So these ring forms in the RBC continue to mature into what we call the schizont, and this schizont contains still multiple merozoites, on average ranging from 8 to 24, depending on the species. So this is the schizont, again formed in the red blood cell, and this schizont undergoes division to form multiple merozoites ranging from 8 to 24. And these merozoites, when they mature within the schizont, this RBC is going to rupture. It is going to undergo hemolysis, and when it ruptures, it releases the merozoites. So the merozoites in the schizont, when the RBC rupture, they are released into blood saturation together with the hemozoids. And because of these toxic substances released, they are the one that causes episodes of malaria attack, like the fever, the headache, the chills, and the sweating due to release of these hemozoids that trigger immune response in the body. So once the ring forms mature into schizons rupture together with RBC, releasing merozoites within the blood saturation. Some of the merozoites, they go and attack new red blood cells. So they attack new RBCs, and after attacking new RBCs, they again form ring forms, then schizons, and the cycle continues. So this is one way of the merozoites. Then other merozoites develop into what we call gametocytes. That is the male and the female gametocyte. So we shall have the male and the female. So we are seeing after the rupture of the RBC, some of the merozoites attack new RBCs and transform into ring forms and mature again into schizons, releasing. So this one helps in increasing the parastemia of the malaria. And we know that parastemia is always very high in PF compared to other species, which show very little parastemia, such as P. malaria. Then some remaining merozoites, for them, they undergo or they are transformed into the gametocytes known as the male and the female gametocyte. And when these gametocytes are formed, both the male and the female gametocytes. So when these gametocytes, so the gametocytes, I'm going to put them in red. So when these gametocytes, the male and the female, are formed in the blood saturation of man when a female Anopheles mosquito is coming to take a blood meal from an infected man. Is going to take up what we call the gametocytes together with the merozoites, but the merozoites do not survive, only the gametocytes survive in the gut of the female Anopheles mosquito. So this one marks the end of erythrocytic schizogony, which occurs in the red blood cell, whereby the ring forms mature into schizons, schizons containing multiple merozoites. Then the red blood cell rupture, releasing merozoites into blood saturation. Some of them attack new RBCs, others develop into male and female gametocyte. That is the end of erythrocytic schizogony. Now we go on the last part, which is sexual, which we call sprogony. And actually the formation of gametocyte, we call it gametogony. Gametogony is the process whereby the merozoites are transformed into the gametocytes. So under sprogony, we are saying it starts when the female Anopheles mosquito is taking a blood meal from an infected human being with gametocytes, and when this mosquito bites man, as it is sucking blood, it sucks up what we call the gametocytes together with the merozoites. And these gametocytes, they move in the female Anopheles mosquito and reach into the gut. And when they reach in the gut, both the male and female, the female gametocyte forms what we call female macrogamete, whereas the male forms what we call the male microgamete. So this one is going to be the microgamete, whereas this is going to be the macrogamete. So the female is transformed into a big macrogamete, whereas the male forms microgamete by the process known as exfragillation. And what is exfragillation? This is whereby the male gametocyte forms flagella by undergoing repetitive division to form what we call flagella. And this flagella enables this male gametocyte to move towards the microgamete so that it can fertilize, or it can fuse with the macrogamete to form what we call a zygote. So don't forget that the male gametocyte undergoes exfragillation, whereby it is a kind of cell division whereby the microgamete develops flagella. And this flagella enables it to become motile so that it can move towards the macrogamete to fuse under fertilization to form what we are calling a zygote. So this is the zygote which has been formed after the motile male microgamete fusing with the female macrogamete, forming a zygote. And once this zygote is formed within the gut of the female Anopheles mosquito, it is transformed into a motile vermicule. And this motile vermicule, we call it ukunate. So it transforms into what we call the ukunate. And this ukunate, you can call it a motile vermicule. This is a motile vermicule which is formed. And this ukunate, because it is sharp, it is able to penetrate the hemocyl of the female Anopheles mosquito. It penetrates the stomach walls and enters into the stomach membranes. And this penetration is done by this ukunate. And when this ukunate reaches into the stomach walls, it transforms into what we call the oocyst. It forms the oocyst within the gut wall. It forms what we call oocyst. This is the oocyst which has been formed. And this oocyst contains multiple sporozoites. These are the sporozoites. So the oocyst contains the sporozoites, which are the infective stage. So the infective stage of plasmodia is the sporozoite. So this is the infective stage. But the diagnostic stage are the trophozoites. So trophozoites are the diagnostic stage. Trophozoites and sometimes schizoids, but majorly the ring forms are the diagnostic stage, whereas the infective stage is the sporozoites. And we are seeing these sporozoites, once they are in the oocyst within the gut wall, they migrate into the salivary gland of the female Anopheles mosquito. So we are going to find multiple sporozoites in the salivary gland of the female Anopheles mosquito. And whenever this infective female Anopheles mosquito again comes to man to take a blood meal, it is going to inject the sporozoites, and then the life cycle continues. So this is what we can discuss under sporogony. Sporogony begins when the female Anopheles mosquito is taking a blood meal from an infected man. And after taking a blood meal, it takes up the gametocytes, both the male and the female, together with the merozoites. But merozoites are killed within the gut wall, whereas the female gametocyte develop into the macrogamete, whereas the male gametocyte undergoes exflagellation, that is developing flagella, forming a male microgamete, which moves and fuses with the female macrogamete, forming a zygote. Then the zygote in the gut wall develops into an ookinete, which is a motile vermicule, penetrates the gut wall, that is the hemocell, and transforms into the oocyst, and this oocyst containing multiple sporozoites, which are the infective stage. And once they reach in the salivary gland, whenever the female infective Anopheles mosquito is taking a blood meal, it injects those sporozoites, hence leading to the continuity of the life cycle. So this is what we can discuss with you in this session. Hope you have enjoyed from this session. Please remember to like this video and share to many others so that they can continue learning with us. May the good Lord bless you.